Hi everyone, this is the third part in our series of lectures on soils and mineral nutrition of plants. We're going to focus this time on nutrient availability, where plants are getting their nutrients, what kinds of factors might control the nutrients that plants have access to. One of the factors on the list was root uptake. Root uptake depletes nutrients near the roots, therefore reducing the overall availability for other plants nearby that might also need to take up nutrients. If we look at this graph here, this graph is plotting distance from the root surface on the x-axis, nutrient concentration in the soil solution along the y-axis. If we look, there are two curves here, one at a low nutrient level here and one at a high nutrient level here. But if we just look at one of these, you can see that as we move farther and farther away from the root, the concentration of nutrients in the soil will go up. Whether we're at a low overall nutrient concentration or at a high overall nutrient concentration, we still see this pattern that they're shaded in pink here. In the two parts of the graph, we see a depletion zone in the area immediately adjacent to the root. So nutrient concentrations decline as we approach the root, they increase as we move away from the root. And this text simply restates that when the rate of nutrient uptake exceeds the rate of replacement in the soil, we see a depletion zone. And any kind of root association, for example, with mycorrhizal fungi would help overcome this problem of nutrient depletion near the root area. So what else controls the nutrients that plants are able to take up? Well, we talked about decomposition rates. But decomposition rates are complex. What's controlling, what kinds of environmental factors or other factors might be influencing the rates at which material decomposes and releases nutrients back into the soil, where they might be available for plant uptake? So take a minute and jot down in your notes any kinds of things that you think might influence rates of decomposition and nutrient availability, and then restart. Okay, so if we're thinking about organic material in the environment, there's a series of factors that you've undoubtedly identified that are either internal to the material itself, these are called intrinsic factors, or that are more related to the environment, extrinsic factors. These would be the internal, these would be the external factors that influence the rates of decomposition. Intrinsic factors would be things like nutrient concentration. When there's a lot of nutrients in organic material, decomposition rate tends to be higher. If we have a very complex litter, lots of lignin or cellulose, the decomposition rate in turn tends to go down, so fewer nutrients might be available in the soil. If we're thinking about external or extrinsic factors, factors like warmer temperatures or greater nutrient concentrations in the environment, or lots of organisms that might be present in the soil, microbes or insects, tend to increase rates of decomposition and make more nutrients available for plant uptake. Greater saturation of the soil can have a couple of different effects, more moisture rather. Moisture in the desert can increase decomposition rates, but moisture that causes saturation, too much waterlogging, can decrease decomposition rates. So there's a lot of factors that might be influencing the rates at which mineral nutrients become released from decomposing plant litter. Here is a figure that shows the relationship specifically with temperature, just one of these factors. If we look at mean annual temperature along the x-axis and the decomposition constant, the rate of decomposition along the y-axis, we see that decomposition rates increase as temperature increases. And this is a pattern that we would typically see. And this, of course, influences nutrients that are available to plants. Other sources of nutrients. We talked about weathering. It was one of the factors that we listed on that earlier slide. Where minerals are tied up in the rock, physical and chemical weathering can result in these nutrients being released. As you can see here, here's potassium tied up in a much more complex structure. If rock is broken down by physical forces like wind or water or temperature, then minerals will be more accessible. Chemical reactions also can release mineral nutrients from where they might be unavailable in the rock. Especially, this is the case when we think about the acids that are released through the decay process. So microbial respiration, plants, fungi, through their cellular respiration process are releasing carbon dioxide. Carbon dioxide can form carbonic acid, which aids in the chemical weathering process. Soil nutrient availability can change with pH and cation concentrations. This figure shows the relationship between pH, here moving from acid to strongly alkaline, or a very low to a very high pH, and the availability of this entire suite of elements. And you can see as you look through here that, for example, if we look at an element like phosphorus here, there are areas where phosphorus is quite available, around a pH of 6.5 to 7, but considerable areas below that, in acidic soils, and also above that in more basic soils, where phosphorus is not particularly available because it binds up to other elements and becomes inaccessible to plants. So the effects of soil conditions in terms of pH can be quite large. Soils can also bind mineral ions through their cation exchange capacity. So if we look here at these particles of clay, vastly enlarged, you can see that there's negative charge sites on the clay particles all around these margins. And these sites of negative charge tend to bind positively charged elements, these cations that you see attached here, the calcium, the aluminum, the ammonium, etc. This occurrence of positively charged mineral ions being bound to negatively charged soil particles is called cation exchange capacity. It's the measure of this absorption of cations. And these cations can be released through a cation exchange process and made available for plant uptake. Here's a schematic that demonstrates that. So if we look here at a root hair that's going to be releasing carbon dioxide because the root is respiring, it's breaking down sugars, it's using oxygen in that process, it's releasing carbon dioxide in cellular respiration. The carbon dioxide that's released in the soil water can become carbonic acid, which you see here, which dissociates and releases these hydrogen ions, which are positively charged, and can then drive the calcium or other cations off of the cation exchange sites. And that's what you see in letter C here. This cation, calcium, has been driven off the soil by these two hydrogen ions, and the calcium then can be taken up by the root. And this is just simply summarized here in the text. Soil texture also influences mineral nutrient availability. Excuse me. As you can see here on this soil triangle, soil texture can vary considerably in its concentrations of clay and sand and silt. There's 2 lab groups who've been investigating the effects of different concentrations of clay, sand, or silt, the composition of the soil, on plant growth. The higher the clay content, the higher the cation exchange capacity typically, and so that's going to influence what minerals might be bound up in the soil or how tightly they're bound and how available they might be to plants. Symbiosis with other organisms, microorganisms, can also increase nutrient availability. This slide's focusing on nitrogen fixation. You can see all the nodules in the roots of this legume. These nodules contain nitrogen-fixing microorganisms that are able to convert nitrogen gas into ammonia, which is a plant-available form of nitrogen. Plants can't use nitrogen gas, but they can use ammonia, which can be fixed from the atmosphere, converted from nitrogen gas by way of lightning or nitrogen-fixing bacteria like rhizobia in symbiosis with a legume, as you can see in this picture, or also blue-green algae or cyanobacteria. Symbiosis with mycorrhizae is also a really key way that plants can obtain nutrients more easily, increase their surface area for searching for elements like phosphorus, for example. You can see in this picture that there's much more vibrant root mass here where the mycorrhizal fungi have colonized this plant. The roots of most plants, in fact, form a symbiotic association with soil fungi, and this permits plants to absorb nutrients, especially phosphorus, more efficiently. The mycorrhizae are getting carbon from the plant, so the plant's photosynthesizing, it's donating some of its carbon to these fungi, and then the fungi are helping the plant to obtain nutrients that might be more difficult to obtain by searching a larger volume of the soil. So it's a true symbiosis. So we've summarized a number of different ways in which plants are influenced by factors that determine nutrient availability in the soil. Soil nutrient availability is influenced by factors such as rates of uptake, decomposition rates in the soil, pH, weathering rates, cation exchange, symbiosis with microorganisms. There are other factors too. But the key point here is to note that mineral nutrient availability in the soil is not a simple process. We tend to think of it as fairly simple because a lot of the nutrients that we think of are derived from fertilizers that we add directly to the soil in the form that plants are most able to take up quickly. But in natural systems, all of these different kinds of processes are what we need to think about when we think about what plants are able to find in terms of nutrients. It helps us to understand why certain nutrients might be more limiting to plant growth in certain areas. We're thinking about a complex interaction among all these different kinds of factors and how they determine what nutrients will or won't be available.